still clarify a point.
Yeah. Okay,
right.
so there is no more questions, so let's move to the last application talk, which is also me. I will have water. And let me share my screen. And this talk is going to be related to Lardan. And the combination of spectral and lifetime will help us in this case. So as I say today in the morning, Lardan is really a wonderful dye. And I will try, if I have time, to make a point of this, because I think this is very important, and there are some confusion in the community about the use of Lardan. And I think this is very important to tell. In this case, what I wanted to do is the understanding of the effect of hydrogen peroxide in the membranes. And for address that problem, we use basically Lardan, and we image our cell using a lifetime microscope and a hyperspectral microscope at the same time. And we took one image after the other in a very short time. So we can consider that the image was taken at the same time in a way. Okay, the lateral organization of membrane is very complicated, because if you think about, we have between 100 to 1,000 of different lipids within the cell. Besides of that, we also have cholesterol, which make our system more complex, as I showed before in the morning. And also we have proteins in the membranes. We have 100 or 1,000 of proteins. And on top of that, we have changes in the polarity or the activity of water due to the molecular crowding. So really, to address the problem of measuring the dynamic of membrane in the cell is a really complex system. And the point that I want to make here is, well, the use of fluorescent proofs that can sense particularly characteristic of the membrane are very, very useful. So one example of this is Lardan, because it can measure the membrane heterogeneity due to the change in the spectra and lifetime, as we saw today in the morning, by the interaction with the particular place where Lardan is at the membrane. So I will not go, I think in the next slide, okay, I will go a little bit in detail again about Lardan, but I will want to stress this thing. Okay, I'm not an expert in oxidative stress. I just know a little bit, but I want to point it out that basically hydrogen peroxide was considered really a bad thing for cells because can produce damage. But in the last 10 years or so, there is a concept that emerged in the biology, which is basically the difference between oxidativity stress and eustress. And these two things are different because one means that is really something that can hurt the cell, but the other are very basically fundamentally for the signaling within the cell. And the cell spends a lot of energy, and it has a lot of system, in simologically speaking, to control the concentration of hydrogen peroxide. Another important thing is the distribution of hydrogen peroxide from outside to the cell to inside to the cell can change in 6,500 or up to 1,000 times between the two places. And it's very interesting that there are some places in the cell where you have a zero amount of hydrogen peroxide. So if you have one molecule, it will make the work. And this is very important. And for this region, you have really a bunch of different enzymes that can reduce with different KDs the hydrogen peroxide. In particular, there is a group of enzymes which they are called Peroxyredoxin. They mentioned in the beginning lecture today in the morning, because there is an important group here, work led by Anna De Nicola, that work in all the reaction of this enzyme with different oxidant that can react really up to the limit of diffusion. There are two models about how this signaling works. One is the flush gate, which means that everything got oxidized, and then when the first enzyme that reacts, the next one can react, and the next one, and this is a path of oxidation. And the other one, which is nowadays more accepted, is the peroxyredoxin sensor, which means that these react really, really fast, and then they transfer basically the information to secondary enzyme to make the work. And we will see that this makes a lot of sense with the result that we have in the paper that we published, because there are two enzymes which are very important, protein kinase C and phospholipase D. So one thing that we knew before we start this was that hydrogen peroxide can change the fluidity of the plasma membrane in yeast. And this produces a reorganization in the supramolecular structure of the membrane, measured by polarization in cuvette using the diphenylhexatriene. So as I said before, we addressed the problem using Laurdan, in which we substitute, the substitution in this point was a Lauril group. And this was designed, as I said before, by Gregorio Weber and Faye Ferris. And I want to make a point here about the importance of this Laurdan, this Lauril group. Well, this Lauril group gives to Laurdan the tension to be a particular region in the membrane in which it can sense a few molecules of water who has restricted mobility in terms of rotation. So if you think in a molecule of water rotating in a glass of water, so the bulk water, it will rotate in the picosecond time scale. However, these molecules are rotating or moving on the nanosecond time scale. And this is very important because this reorganization of the water molecule around the dipole is competing basically with the emission of the Laurdan. And this is what we can sense when we analyze the dipolar relaxation. So if we can measure the lifetime in two channels, one in the blue and one in the red, we can split these two parameters, which are basically the polarity and the dipolar relaxation. And the polarity is related to the average dielectric contrast of the apparent dielectric contrast that we have in the environment of Laurdan, and the dipolar relaxation is the physical reorganization of the water around the probe. So this was basically what I was trying to illustrate. And the point that I want to make is, if you modify the Laurdan with any kind of different structure, this will move Laurdan up and down in the location in the membrane, and at the end you will be sensing different things. And it's not possible to tell that you have similar proof of Laurdan basically because you have the same chromophore, without taking into account that basically the location of Laurdan is due to basically the fluorophore, but also the Lauril group. And this is important because otherwise you will be sensing different things. And as soon as you change also the chromophore, it will change the distribution of Laurdan depending on the liquid order, disorder, or phase membrane. And Laurdan, one of the unique things is that it can diffuse across all the membrane without have any particular affinity for each of these. So I already went there. We will use basically this thing of measuring FLIM and hyperspectral, and I explained how this works. So we will use NAH fibroblast, and then we will place it, wait for 24 hours, and then we will stimulate using a concentration, a bolus of hydrogen peroxide in 2 concentration, and wait for an hour and do the measurement for our experiment. So how we will analyze the data? So we will have 3 variables. So we will have one set of data, which is FLIM, and the other will be spectrum. So the FLIM, we split in 2 channels. So for the channel 1, that I call blue, is the range in the more or less center in 440. We will collect our data, and then we will analyze using this 3-component analysis in order to see the change in the fraction of the liquid order and liquid disorder, and then if there are dipolar relaxation. we will not expect to have dipolar relaxation in the blue channel. We will have to see dipolar relaxation in the green channel because it's related to the environment in the water. So the relaxation of water across the probe. So for this, we will basically trace what is the fraction of dipolar relaxation regarding one dimension, and what is the fraction of fluidity regarding regardless of the relaxation or not. And this is how we analyze the data, and this was introduced by Suman today. So if we do this and we treat our sample with hydrogen peroxide, what we immediately see is analyzing the full image, we didn't notice much difference. We see that there is a decrease basically in the distribution. And this distribution from here is basically the histogram of all the pixels along this line, right? Then we calculate the center of mass of this distribution, and we compare the center of mass for each of the cells between the two groups. And this is what I am presenting here. So we did the same for the dipolar relaxation. And what surprised me immediately was that we do have a change in the dipolar relaxation in the blue channel. And this was unexpected. Let's keep this in mind for the discussion at the end. When we analyzed this, but now using mask, everything became more clear. So when we do mask, we only focus our eyes to this region. And in that case, you can see changes in the membrane, and the changes are moving toward to less fluidity. So the membrane are becoming more ordered. The same happened with the dipolar relaxation. So the dipolar relaxation basically is increased when we have more ordered membrane, which is in a way anti-intuitive. So then we analyze the second channel. The second channel was the green, and this will be focused mainly in the move out of the universal cycle because it's a reaction during the excited state. So it will pull the data out of the universal cycle. Something similar happened, but in that case, you can see there is another component here, and when we analyze the center of mass, we do see a difference, significant difference between these 2 points. And this difference is huge when you compare the dipolar relaxation. So it's really extreme change toward to higher relaxation when we have hydrogen peroxide. When we reanalyze the data using the same logic, and basically we segmented and we collect the region where are more related to plasma membrane, this also happened again, and we can see a shift back to less fluidity in the plasma membrane. And then the most important thing here is the dipolar relaxation, and the same happened that in the whole cell analysis. But now we have also a trend by the different concentration of hydrogen peroxide. So as a bottom line, fluidity decreased as dipolar relaxation increased by hydrogen peroxide in the green channel. So, but as I said before, we have also spectra, and we collect the spectra, and in the spectra, we basically have the 2 variables, the fluidity or the order disorder and the dipolar relaxation together, moving along this line. In that case, we have a strong background. And as I said before, this is not a problem. You need to include in the system and then trace back where should be the position of the pixel in our linear combination. When we do this and we analyze the plasma membrane, you can see that there is a shift toward to increase in liquid order, which is in coincidence with what we get for the lifetime when we introduce the hydrogen peroxide in the system. And this is also significant when we analyze the center of mass of the distribution along this line. So finally, what we did was trying to connect the dimension of the lifetime with the dimension of the spectral phasor in order to see if we can figure it out what was going on between this spectral shift, the dipolar relaxation in the blue and in the red channel. So in the analysis, what we did, we placed cursors here, and these cursors were fixed. This is the linear combination for liquid order, liquid disorder, regardless of the channel what we were analyzing. And this is control and hydrogen peroxide. And then we did another linear combination for the dipolar relaxation. When we analyzed, when we found this pixel here, then we tried to trace back where was those pixels in the lifetime channel 1 and in the spectral phasor. And you may notice that the shorter lifetime here and here are the shorter lifetime in the other channel, and also the redder pixels in the spectral phase. And the other side is also true. The green here are more toward to the plasma membrane, you can see here, and also toward to the longer lifetime here, you can see here better, right? And then it's moved toward to the liquid order here. In the dipolar relaxation, you may see that the most outside is related to the interior of the cell, right, in the both cases, and then is here on the universal cycle, and here a little bit outside on the universal cycle, and the same is true for the other case. So at that point, I was absolutely lost, and I came back as usual to the Enrico's office and say, Enrico, I cannot figure it out why we have dipolar relaxation in the blue channel. And as usual, Enrico came up with a very nice idea, which was, well, he already did some experiment with Tiziana Parasasi in this paper very early in Cuvette, the 1993, in which if you have fast interconversion in the system, which means if you have a domain, basically the time that take the Laurdan to go from one to another will basically give you a final result that if you get excited in one environment, it will emit in this environment. It will not change. You will have on average not change from one system to another. Remember that the diffusion of Laurdan is very fast, more or less in 1 micrometer square per second, and this allows you to travel that distance like this. Okay, if this is true, what he pointed out is, well, what happened if you have structure which are like filaments in the membrane. And this happened, for example, in shell membranes. And they demonstrate in this situation that when you have shell membrane, you can have excitation during the shell, but then the emission in the other membrane. And if this happens, you will have this effect of basically exciting in the blue, but emitting in the relaxed environment. And at the end, you will have relaxation in the blue channel. I found basically this really nice explanation for the result, but now we have to explain this from the biology. And the explanation that I found is this thing. Well, hydrogen peroxide can activate protein kinase C, and protein kinase C is activated by, well, phospholipase D can activate, it can be activated by protein kinase C. And the phospholipase D is in charge of chopping basically the head of the phospholipid, making a phosphatidic acid. And this changed quite a lot the mental transition of the phospholipid, making completely solid at the temperature where we ran the experiment. So in that case, what we hypothesized is that if we have this situation, I did some more experiment. I never finished this experiment in order to corroborate this hypothesis, but we did experiment with a sensor for protein kinase D, and we were trying to stimulate and basically inactivate PLD or inactivate PSC in order to see if the stimulation of hydrogen peroxide can reproduce the experiment that we did for Laurdan. Still not conclusive, but basically if we have lines that the Laurdan can get excited here, but then moving out, this is the explanation that we found for this application. And the interesting thing here is, well, basically using the best of each of the both methods, we can conclude something which is very difficult to interpret and to give us a solution, which is very, very, very important. So this is finally the end of my talk.